Welcome back, Bio30 Prep. We're going to continue our discussion on human body systems, starting with the digestive system. In this lecture, we'll be looking at macromolecules. In your textbook, this is on pages 200 to 216. By the end of this lecture, you'll be able to define the term macromolecule, identify the role macromolecules play in living organisms, describe the chemical structures of carbohydrates and lipids, explain how synthesis and hydrolysis of carbohydrates and lipids occur in general terms. Here we go. So when we look at the human body, this is kind of a schematic of the human body where the human body is composed of three parts: cytoplasm, blood, which is, I guess, how things are transported outside of cells, and then all the volume between all the body cells. So most of our body is cytoplasm. It's cells contained inside cell membranes. There's a little bit of fluid stored outside of them in what's called interstitial fluid. And then there's the volume of our blood plasma. If we take a look at these things, what we need to see is that the body consists of more than 60% water spread throughout these three divisions of the body. Water, small molecules, and ions can move between these areas by osmosis and diffusion. So small molecules can pretty much move around throughout our body by diffusion, maintaining a kind of a constant concentration of these small molecules and ions. Large molecules, macromolecules, can't. They're too big to fit through cell membranes. They're too big to even fit. If you look at our circulatory system, this is a capillary wall, and if you'll take a look, it's only a single cell thick. This one's showing 3 cells, but what we really should be seeing is that the wall itself is only a single cell thick. Large molecules can't pass through where the cells join. So these macromolecules, what are macromolecules? Well, organic macromolecules fall into these categories. We talked about them a bit in the last lecture. Carbohydrates, proteins, lipids, nucleic acids, those are all macromolecules found in the cell. They're organic molecules in that they always contain carbon and hydrogen. So if a compound contains both of these elements, it's an organic molecule. A macromolecule, simply, if we take a look at the word here, macro means big. And so that's exactly what macromolecules are. They're big molecules. They're thousands of atoms in size, or can be thousands of atoms in size. The way they're made is, just as atoms can join to form a molecule, molecules can then join to form macromolecules. So they're just really joining of a whole bunch of molecules into one single large molecule. When it's the same type of molecule, a monomer, so when we're looking at macromolecules, if they have a consistent unit that's being repeated, or a monomer, the macromolecule can then be called a polymer, poly meaning many, or many units that are the same in that compound. All of our organic molecules are polymers. So carbohydrates, proteins, lipids, and nucleic acids are all simply repetitions of a single base unit over a long period of space to create a large molecule. Here's an overview of what organic macromolecules do in our body. Carbohydrates and fats and lipids are the same thing. Their primary role is energy. Carbohydrates are easily accessed energy. Fats is more of a storage form of energy. They're also used in cell membranes and to transport vitamins. They're also involved in some of our hormones. But the biggest thing we should remember is that fat is an energy storage molecule, and sugar or carbohydrates, same thing. In terms of the subunits, carbohydrates are just sugar units linked together. And there's many different types of single sugar units, but the base unit of all carbohydrates is simply a sugar. Fats have 2 base units, or 2 subunits: fatty acids, which are really just carbon chains for the most part, attached to a glycerol molecule. And I'll take a look at one of those right away. Or another form is the phospholipid that you see in a cell wall. Proteins and nucleic acids, on the other hand, have a much different function. So the fats and the carbohydrates, you can kind of group them together and you can say, well, this is energy. These are the molecules our body needs for energy. That's the big general view of carbohydrates and fats. Proteins, on the other hand, their subunits are called amino acids, and they have two roles. One is a structural role, where they're components of hair and fingernails. The other one is what I call a machine role. They do things. So, for example, transporting oxygen, clotting our blood, chemical digestion, all of these things are done by protein molecules. Some example protein molecules: keratin. This is what your hair and fingernails are made of. Hemoglobin. We all know that's in our blood. Pepsin and amylase are digestive enzymes. Fibrin is used in blood clotting. And actin and troponin are actually proteins in muscle contraction. So every time there's a job to be done in our body, it's being done by a protein. Nucleic acids, on the other hand, are made up of things called nucleotides. And one thing I'll point out here is you really want to get these two things, the amino acids and the nucleic acids, separated in your mind. Proteins are made of amino acids. Nucleic acids are something entirely different. They're made up of nucleotides. Another way to think about this is nucleic acids are a macromolecule. The amino acids are a subunit. And you want to get those two things sorted out, otherwise you'll end up saying nucleic acid when you mean to say amino acid. And I'll probably do the same thing on occasion, but we really want to get those two separated and have a clear idea as to what their differences are. In terms of nucleic acid, this is our DNA and RNA, or our genetic information. So a much different role from proteins and a much different role from fats and carbohydrates. So those are the four major molecules in our body. Each one has a specific task or a specific role. When it comes to reactions involving macromolecules, in terms of making them and breaking them down, the reactions are actually quite simple. Every single macromolecule, despite the fact that there's a large variety of subunits, undergoes a very similar reaction when it's being built or when it's being disassembled. So when we are building macromolecules, every single one of those macromolecules, whether it's carbohydrates, lipids, proteins, or nucleic acids, have hydrogens on one end and hydroxide groups or hydroxyl groups on the other. These three atoms can pop off of any of these subunits to become a water molecule. When they do, they leave behind an area where bonding can occur. So it's called dehydration synthesis. Synthesis because something's being made or building. Dehydration because we're removing water. So by removing that water molecule, or by removing those three atoms to make a water molecule, we're able to connect the subunits of our macromolecule. When we want to break macromolecules, and we do this when we do digestion, so for example, this would happen in this kind of reaction is digestion. Digestion is really the process of adding water to a bond in order to break it. When we break it, a hydrogen ends up on one side of our macromolecule, and a hydroxide group or hydroxyl group ends up on the other. And what this does here is, now look, we have a subunit. And this can now be absorbed by the body. It can be absorbed into our circulatory system, carried out to a cell, and that cell is going to use it to build its own macromolecule. So two big reactions. Dehydration synthesis is the reaction that's used to make macromolecules. Hydrolysis, or I think hydrolysis, because lysis means to break, is the reaction that breaks macromolecules back into their subunits. Both reactions are completed using enzymes, and in the next lecture we're going to talk about what exactly are these things called enzymes. So for carbohydrates, they contain carbon, hydrogen, and oxygen atoms. So they only contain 3 atoms total, or 3 types of atoms. That's it. If it's a single sugar, or if it's a simple sugar, it's going to have a ratio of 1 to 2 to 1 in terms of carbons, hydrogens, and oxygens. It's life's most important source of energy, and this is what plants make when they do photosynthesis. When plants do photosynthesis, they make their own carbohydrates, they make glucose, and the glucose can be used to make a carbohydrate. Animals, on the other hand, rely on plants for their carbohydrates. So we have to either eat a plant or eat another animal in order to gain the sugars in their body and do cellular respiration. Now, simple sugars aren't the only form in which carbohydrates come in. As they get linked together, the terminology, or their names change. So if it's a simple sugar, if 2 of these simple sugar units are joined together, it's called a disaccharide. Disaccharide because 2 joined together. Once you get 3 or more joined together, it's called a polysaccharide. So some examples of some sugars. Often sugars end with an ose ending. Think of sucrose, glucose, fructose. Those are ones you've probably heard before. Fructose is what you're going to find in soda pop. Glucose is what you'd find in maple syrup. And sucrose is what you put in your coffee, if you drink coffee with sugar, that is. And what you'll notice is that all of these simple sugars, or these 2, follow the basic 1 to 2 to 1 ratio. So 3 to 6 to 3, there's twice as many hydrogens as there are carbons and oxygens. Same thing with hexose, twice as many hydrogens as there are carbons and oxygens. Once that ratio changes, so if we take a look at sucrose here, sucrose has twice as many hydrogens as oxygens, but not carbons. So that ratio doesn't work anymore. And so this is a disaccharide. This particular one is formed by joining glucose and fructose by dehydration synthesis. And if we take a look, if you take a C6H12O6, so there's a glucose, and we add a C6H12O6, so there's 2 single sugar units going together. When they react, we know that there's 1 water molecule formed, and the rest of those atoms are joined together in a molecule. And if you remember your conservation of mass, we know that these atoms can't disappear, so they have to be, the number of atoms on one side of our equation has to be the same as the number of each type of atom on the other side of our equation. And if we take a look, we have 1 oxygen over here, which means the rest of them have to be on our sugar unit. And if we count them up, there are 12 total on the reactant side. 12 minus 1 is 11. If we take a look at our hydrogens, we have to have 24 total, 12 from each of our sugar units. 2 of them are here, which means there's 22 hydrogens left over. And then for carbons, 6 from each of our single sugar units. And so there's our final molecule. And as you can see, when we put these 2 sugar units together, it's because we lose that water that our ratio changes. Both plants and animals make their own carbohydrates. In animals, it's called glycogen. It's a storage unit for energy in our body. It's a polymer. It has a similar structure to starch. Cellulose and starch are the plant versions of carbohydrates. Cellulose is used to make up cell walls. Starch is a polysaccharide that's purpose is energy storage, but cellulose, its actual purpose is cell walls, so it's a structural carbohydrate. Monosaccharides, it's another name for simple sugars. Single unit sugars, glucose and fructose are the most common. They have the same chemical formula, but different chemical arrangements. So they're isomers. They have exactly the same chemical formula. If you take a look at these two pictures of fructose and glucose, you'll see they're the same, except at the end here, there's an oxygen, and I should put these in, but in chemistry, anytime there's an intersection or a bend in a chemical, it means there's just a carbon atom there. So there's carbon atoms on all these spots here. And if you add up all the atoms, there's the same number of carbons and hydrogens and oxygens on both molecules. The only difference is the placement of this double bond. In fructose, the double bond is on the second from the end. On glucose, it's on the very end, where that double bond with the oxygen happens. These structural differences actually make fructose about 1,000 times sweeter, and that's why it's the sugar that's used in pop, is because you need less of it to make it taste sweet. So even though same chemical structure, shape affects its chemical properties. Here's just a slide showing that same kind of reaction. It turns out that on the last slide I showed these molecules in a linear arrangement. Most of the time glucose is in a circular arrangement. Each one of these points on our hexagon is a carbon atom, and at the end of each is a OH and a H. So here, joining two glucoses together, or two monosaccharides together, when they're being joined, it's dehydration synthesis for making that water molecule, and they're bonded across an oxygen. So this is kind of the bridge between two single sugar units. And since there's an O and an OH over here, we can always add another sugar, and then another sugar, and then another sugar to make a polymer. So dehydration synthesis is the removal of a water molecule. When two units join, the hydroxyl group is removed from one subunit, and the hydrogen is removed from the other. When we start tearing molecules apart, it's called a hydrolysis reaction. It does the exact opposite. A water molecule is going to come in and attack this bond and separate it. Example disaccharides, here we have lactose. So again, these things can have the same chemical formula but different shapes, which gives them different chemical properties. So these are all isomers of C12H22O11. The most common one that we're aware of is sucrose. It's our table sugar, formed by plants, and excessive exposure to this stuff can lead to type 2 diabetes. It's also one of the sweetest, in terms of taste, disaccharides. As we go down our list, maltose has the same basic shape. I don't have a picture of it here, but there's probably one in your textbook. It's also produced by cereal plants, so grains. It's a lot less sweet than sucrose, but sweeter than lactose. And lactose is the sugar found in milk. When we compare these three, it's the least sweet, even though it has exactly the same chemical formula as the other two. And it's when you have lactose intolerance, it's because you're missing an enzyme that's able to hydrolyze this bond. So when we break down these disaccharides, what we're really doing is we're breaking this bond here by adding water. If you're missing an enzyme, which is really just a machine that's going to help this happen, that's when you're going to be lactose intolerant and get cramps. You're not going to be able to break down this bond, and as a consequence, these disaccharides are going to be moving through your bowels, and your bowels want to break them down, so it actually slows down to give it more time, and that's what causes the cramping and the other discomforts associated with lactose intolerance. When we look at the big polysaccharides, Animal polysaccharides, which you're going to notice in terms of structure, is they form these big branching structures of glucose subunits. The big difference between animal polysaccharides and plant polysaccharides is shape. Animal polysaccharides branch more, which means they can pack more densely. We can store more sugar in less space. So glycogen, that's one word we should remember. It's our energy storage molecule. Those glucose subunits are brought to the liver and assembled by the liver and muscles by dehydration synthesis, and then they're stored as glycogen. When the sugar levels in our blood drop, the reverse reaction happens, and we get hydrolysis. So those are animal polysaccharides. Plant polysaccharides, notice a much different shape. Here we have a starch molecule, long, not very many branches, still glucose subunits. When we look at cellulose, this is a structural polysaccharide, so it's there to make things strong, and you'll notice it looks kind of net-like our web light, where we have these single sugar units joined in several different directions. So generally what happens is disaccharides leave from the leaves after photosynthesis, and they're transported to the roots of plants where the polysaccharide is then formed. So starch is the same idea as glycogen. Cellulose makes up cell walls. Its unique structure makes it really hard for us to digest, and this is really, when they're talking about fiber, like dietary fiber, what they're talking about mostly is cellulose. We can't digest it, but what it does is it adds bulk to our bowels, and it helps keep our bowels clean. So by eating all that lettuce and high fiber food, it bulks up it almost scrubs our small intestine out and makes it so that toxins are less concentrated and that kind of thing. So that's why fiber is important, even though we can't get the energy out of it. And if you take a look, we mentioned a few lectures back now that hydrogen bonds would be important. Here's one mention of them right now. It turns out that these chains are attracted to each other by hydrogen bonds, these cellulose chains, and that's what gives them strength to form structures of leaves and stems. Now moving on to lipids, probably one of the most important lipids we'll talk about are triglycerides. They're composed of glycerol and 3 fatty acids. There's phospholipids. We've already talked about this quite a bit. It's composed of a glycerol, 2 fatty acids, and a phosphate molecule, and that phosphate is the hydrophilic head. There's also waxes. These are long chains of fatty acids joined to alcohol chains or carbon rings. We won't spend much time talking about waxes. If we take a look at our structure here, let's just make sure I get the circle in the right place. This here is our glycerol molecule. So carbons go there. So there's glycerol. Each one of these is a fatty acid. Where an R represents a carbon chain. And it turns out that each chain can be different. That's why there's a 1 and a 2 and a 3. 3 different, or they could be the same, but there's nothing wrong with them being different. 3 different carbon chains attached to a glycerol molecule, and what this does is it's used to store energy for a long time, and also it's almost like better packaging. Fats can store way more energy in much less space when you compare them to a carbohydrate. So most are used for energy storage. They're also cushioning and insulators against the cold. Carriers of some vitamins such as A, D, E, and K. They're also the raw material for some hormones. 3 major types we mentioned already. This is why we want to talk about triglycerides, and this is the one we're going to focus on in this digestion unit, are the triglycerides, because we're talking about the energy, and that's what we want to get, that's what we want to make when we're doing digestion. Here's another picture of a triglyceride, a little more expanded out. And if you take a look, here's our glycerol molecule. Notice I circled in that last diagram 3 oxygens and 3 carbons. The hydrogens weren't shown. And then same reaction. Look at this. There's an H. There's an OH. That's going to become a water molecule. Here's the water molecules over here. That's dehydration synthesis. So to make triglycerides, we simply do 3 dehydration synthesis reactions. And then you get this fat that stores lots of energy. So glycerol and fatty acids form through dehydration synthesis. 2 types of fats. Saturated fats means that only single bonds join the carbons in the fat. So if we take a look at this picture here, this particular chain, this would be a saturated chain. Because if you look in between the carbon atoms, there's only a single bond. And what this means is each bond represents the sharing of electrons, the sharing of 2 electrons. These carbon atoms are only sharing 2 electrons each. It means they're stable. They're not, electrons, remember, are negatively charged. So there's only 2 in between each carbon atom. That's okay. The carbons are happy. Unsaturated fats, and so stable. Stable means they form solids or semi-solids at room temperature. If they're stable, they're attracted to each other, and they don't have that much energy, so they stick together. Oils, however, are the triglycerides and fats, and these are our unsaturated fats. So if we take a look at this particular fat chain, here we see that double bond. This is unsaturated. What that means is if we broke this bond, we could put more hydrogens into our fat molecule. So unsaturated or polyunsaturated would mean there's more than one, poly just meaning many. That there are some double bonds existing between the carbon atoms. This makes the lipid more reactive than the animal fats, which means they're easier to break down. There's now a spot to attack on the molecule and break it into smaller pieces. There's more tension in that spot because now there's 4 electrons being shared between 2 molecules, 4 negatively charged particles being forced between 2 molecules. Less stable because negative charges are all pushing against each other. So as a result, because they have a little bit more energy in them, they're liquids at room temperature. Think of your olive oil, palm oil, sunflower oil. All these oils that we use in cooking are plant oils. Trans fats, these are unsaturated fats, so they sound like they should be good for us, that have been partially hydrogenated in the lab. Hydrogenated means add hydrogen. And what this does is it makes the compound more stable, and in terms of food, it gives food a longer shelf life when it's sitting on the shelf in a box. So what happens is it removes the carbon-carbon double bonds, making these fats more stable. If we take a look at some fats, and so these are fat chains, or some fatty acid chains, unsaturated fatty acid chains. So now I got to get my pen here. Here we go. Here's our double bond of carbons. The black circles are the carbons, the white circles are hydrogens, the red circles are oxygens. But if we take a look at this unsaturated fat, we have a bend in the molecule. This makes it really easy to attack it with water molecules and break it into smaller pieces. So we end up with one piece here, one piece here. These are more easily absorbed by the body. If we take a look at a saturated fat like the one found in milk, notice this long straight chain. There's no easy spot for water to get at, so these things don't break. And these long chains end up as plaque in arteries. This is why animal fats harden your arteries. It's because these long molecules, they don't break because they're so stable, and then they build up, they get lodged together inside our arteries and literally start building plaque, making our arteries narrower and thereby increasing our blood pressure. If we move over to our trans fats, now look, if you take a look, here's where the difference is. And it turns out it's still a double bond. However, it looks like our saturated fat. So now we have this molecule that is long, straight, won't break down, and now it's leading to the same problems that those saturated fats are causing inside our circulatory system. So that's why trans fats are bad for you. Even though they're unsaturated, they're made to look saturated, making it harder for the body to deal with. So in summary from this lecture, what you should get is that macromolecules consist of subunits that are joined by dehydration synthesis. They can be separated by hydrolysis. Those are the two reactions we're going to be talking about throughout the digestion chapter. Carbohydrates are energy-storing molecules. They're classified based on the number of simple sugar units they contain. There's your monosaccharides, disaccharides, polysaccharides. And we should also know that there's glycogen and starch. Glycogen is especially important because we'll be talking about it both in Bio 20 and in Bio 30. So the idea that glycogen is a polysaccharide there we go. Polysaccharide is important in terms of both Bio 30 prep and Bio 30. Lipids are primarily an energy storage compound as well. They also form the cell wall of cells, and they can also be used to transport vitamins. In the next lecture, the one right after this, we'll talk more about proteins and nucleic acids. And we'll finally get to what exactly an enzyme is.